Hello, I'm going to talk about my research involving the formation of Staphylococcus aureus persister cells in the long-term survival phase and destruction by a combination of conventional antibiotics. I did this research in the Department of Food Science at Penn State in a foodborne pathogen lab under Dr. Stephen Knabel. So let's get started. Persister cells are defined as a small subpopulation of bacteria that are tolerant to antibiotics. Tolerance to antibiotics is thought to be due to persister cells being dormant, characterized by a minimal to no metabolic activity and a shutdown of replication. It has been demonstrated that in the Knabel lab that the frequency of persister cells reaches 100% in the long-term survival phase in Listeria monocytogenes. To determine if Staphylococcus aureus reaches 100% persister cells in the long-term survival phase also, penicillin was used as a treatment at various phases of growth. Staph aureus persister cells were then exposed to the antibiotics penicillin, rifampicin, gentamicin, and a combination of rifampicin and gentamicin to evaluate the effect of different combinations of antibiotics in the destruction of Staph aureus persister cells. The destruction of dormant persister cells by a combination of rifampicin and gentamicin suggests a low level of metabolic activity is still occurring within persister cells during the long-term survival phase. We speculate this minimal metabolic activity may allow long-term survival via the synthesis of critical survival proteins. The formation of persister cells involves the stringent response and toxin-antitoxin interactions. Activation of toxin-antitoxin systems is mediated by the alarmone guanosine tetra and pentaphosphate, termed PPGPP, shown in red, whose synthesis is catalyzed by the enzymes RelA and SPO-T. The alarmone PPGPP is capable of both degrading and acting as a PPGPP synthase. So the PPGPP inhibits the poly-P degrading enzyme PPX. Poly-P is an enzyme that is regularly produced by a polyphosphate kinase, also called PPK. The poly-P stimulates LON protease, and the combination of poly-P and LON degrade antitoxins. You can see that coming together in the figure. This combination has been found to degrade 11 antitoxins in E. coli. With the degradation of antitoxins, toxins are left free to inhibit translation and therefore arrest cell growth. In this figure, the arrow pointing to the TA promoter from the toxin indicates the abundance of free toxin that maintains dormant cells in the persister state. This is the system which is thought to mediate the formation and maintenance of persister cells. Persister cells were initially discovered as a result of a study on the mechanism of action of penicillin. Joseph Bigger noticed in 1944 that a population of non-penicillin-resistant Staph aureus developed the ability to survive penicillin exposure. Penicillin can be used to determine the frequency of persister cells present in a culture because it targets a process, cell wall synthesis, that is inactivated during dormancy. When persister cells are dormant, they are not forming new cell walls. Therefore, any non-replicating cells survive exposure to penicillin and can be identified as the persister cell frequency within a given culture. This concept is demonstrated graphically. The addition of penicillin to Staph aureus results in the killing of non-persister cells and gives you an idea of the quantity of surviving persister cells. The arrow on this figure demonstrates that persister cells can then grow and become a susceptible population once again. This experiment involved exposing 100-hour-old persister cells to a variety of antibiotics. This figure walks through the formation of persister cells. At 0 hours of incubation, all cells are vegetative. This continues through log phase. By stationary phase, there is a deceleration and stringent response, which leads to a small subpopulation of persister cells. Days and months lead to the formation of long-term survival cells, a 100% persister cell population of Staph aureus. This is the population I exposed to antibiotics for an extended period of time. I chose penicillin as my standard, knowing that it would always be 100% persister cells no matter how long I exposed the culture to antibiotics. I also used antibiotics gentamicin and rifampicin individually as well as in combination to treat the persister cells in hopes of killing them. The figure demonstrates the range of sensitivity I anticipated. I did not expect the persister cells to be sensitive to penicillin. I anticipated moderate sensitivity to gentamicin and rifampicin, and I was hoping to find high sensitivity to the combination of gentamicin and rifampicin. Antibiotic exposure was extended for up to 6 days, and samples were taken daily to evaluate the survival of persister cells in the same manner. This experiment confirmed that previous findings that Staph aureus persister cells have no sensitivity to penicillin. Cell wall synthesis was likely not occurring as a result of dormancy and resulted in subsequent shutdown of reproduction. There was a slight sensitivity to gentamicin as translation was occurring and therefore targeted by the antibiotic. At 4 days of incubation under the stress of exposure to gentamicin, complete tolerance was observed within the long-term survival culture. Data also suggests that there was slight sensitivity to rifampicin, indicating transcription was still occurring. At 3 days of incubation under the additional stress of exposure to rifampicin, tolerance was observed within the culture. Persister cells showed significant destruction in the presence of a combination of gentamicin and rifampicin. The data suggests a synergistic destructive effect of this combination of persister cells. Destruction was likely due to the failure to synthesize survival proteins in the presence of these two antibiotics. Each experiment was repeated twice, and error bars indicate the standard deviations about the mean. An asterisk denotes data from only one replication of the experiment. When used in combination, rifampicin, a transcription inhibitor, and gentamicin, a translation inhibitor, had a synergistic effect and successfully killed Staph aureus persister cells in vitro. This may be due to the lack of synthesis of survival proteins that may be needed for the long-term survival of persister cells. Understanding ways to kill persister cells is paramount to developing appropriate antimicrobial therapies for curing the many devastating chronic infections that are currently causing a high rate of morbidity and mortality throughout the world. In the future, lower concentrations of antibiotic treatments should be explored to identify combination therapies that can be used to cure chronic infections in humans. Additionally, further experimentation should be conducted to determine whether or not survival proteins are responsible for maintenance of the long-term survival phase in persister cells. Thank you for listening to my presentation, and thank you also to my research mentor, Dr. Stephen Knabel, and graduate student Matthew Doan, for helping me extensively with this research project.